This study started 5 years ago in 2017 when a patient that was diagnosed of breast cancer asked to us if she could had transmitted the breast cancer to a baby through the breastfeeding. So we explained that this is not the case, but anyway, she provided to us a breast milk sample collected 18 months before the breast cancer diagnosis, and then we were able to analyze that sample and find the same mutations that were present in the tumor. The method that we are using for patients during the postpartum are only the study of patients with symptoms. In this study, we analyzed the utility of an NGS panel that we built at BIO with Ana Vivancos at Genomics Lab, including the 54 mutations that are more common in women diagnosed of breast cancer with less than 45 years old. And we are working on our next step of the project. It's to test if this NGS panel could be useful for early detection using the breast milk. So the results that have been published in Cancer Discovery are summarizing the results of 12 controls of patients, healthy patients that provided to us breast milk, and all the samples were negative for those patients without cancer and at least a 12 months follow-up. And we also present the results of 15 patients with breast milk samples collected during the breastfeeding in women diagnosed during pregnancy or in the postpartum. So for those 15 patients, we were able to detect the mutations in the breast milk in 13 of those 15 patients using that panel. And the 2 patients that were negative were 2 patients with the samples collected just after the delivery, less than 5 years after the delivery. And we may hypothesize that perhaps the samples are negative because in the colostrum, we do not have enough time to deliver the ctDNA in the breast milk. But those 2 patients had to stop breastfeeding to continue with the breast cancer treatment. As of today, the results are very nice for us from the scientific perspective, but now we have to recruit 5,000 women to see if the NGS panel that we built is useful for early breast cancer detection. So we are now in the stage of looking for funding for this trial, and our plan is to collect worldwide 5,000 women that are pregnant, at least with 40 years old, or are mutation carriers of BRCA1 or 2, PALB2, or RAD51C or D, and then apply this NGS panel that we built at BIO, and we will analyze the breast milk samples, the blood, and we will perform ultrasounds and mammographies during 2 years for those patients. And then we will be able to see if our NGS panel is better than the other methods to detect early breast cancer.